Alright guys, today we're going to talk about embryogenesis and the formation and use of endosperm. Now this is basically the formation of the seeds. So up to now we've talked about the formation of the male gametophyte, which leads to the pollen grain formation and the formation of sperm. We've talked about the formation of the female gametophyte, excuse me, which leads to the formation of the female, which is located within the ovule and leads to the formation of the egg. We talked about pollination and double fertilization. And immediately after double fertilization is where we are going to pick up. And that's the formation of the zygote, which eventually develops into the embryo, which eventually develops into the mature sporophyte. Now, in double fertilization, we don't just produce the embryo, we also produce a really unique tissue in the entire tree of life. Nothing else is like endosperm. It's a triploid nutritive tissue, and it's not the same organism as the zygote. It's kind of like the sister to the zygote from a genetic standpoint. And we'll talk a little bit about the evolutionary implications of that at the end of this lecture. So endosperm may be persistent, like in rice or corn, where you have very small cotyledons in the mature seed, and the endosperm, in grains at least, is what you eat. And it acts as, once the plant starts to germinate, that's when the tissue is consumed. In most dicots, like the bean, the endosperm is absorbed very early in development by the cotyledons. And so the large fleshy part of the bean is actually the cotyledon or the seed leaves. And what you see here, these first little leaves, are actually the first true leaves. And these are known as plumules. The epicotyl, it looks like it's pointing to the plumules in this picture where it's pointing to the epicotyl, but the epicotyl is actually a small stem-like region that's above the plumules. Okay, so it's kind of hard to see there because it's being protected by those plumules. The epicotyl is. All right. So when you talk about embryogenesis, you can talk about the different stages of it from different points of view. So if you look at it based on the developmental events, there are two basic phases. And the first one is the morphological events or the morphogenic events, and that's when the basic body plan is being determined. And the morphogenic events occur very early in embryogenesis. So the globular phase to the heart phase. So these are the morphogenic events. Then once you get this sort of bilateral symmetry, all of the major organs, all of the major tissues, the patterning, both the radial patterning and the longitudinal axis, and both of the primary meristems have been developed. And so after this phase, after this heart phase, you move on to what's known as the post-embryonic development, and that's the latter phases of embryogenesis when all of those tissues get bigger, the cells divide, and they store nutrient tissue to get ready for germination. So the first phase here, the morphogenic events, determine the different parts of the plant body, and then the latter phases, the late heart phase, torpedo phase, walking stick phase, these are all growth and nutrient storage, and then the establishment of dormancy. Now, so that was based on a developmental patterning. Those were two basic phases, morphogenic events and post-embryonic development. But you can also look at the embryogenesis, or the formation of the seed, in terms of its morphological forms. And so when you look at it when you classify it based on morphology, you see four major stages. The first is the globular phase, which are these first three images, and that's when the embryo, the embryo is globular. It's radially symmetrical. It looks like a circle. The heart stage is when you establish bilateral symmetry. Okay, now notice here, this is well into the heart stage, but the lobes of the heart are pointing outwards. Okay, here the lobes of the heart are pointing outwards. When you get to the torpedo phase, as you see here, these lobes begin to point inwards. And if you look at some of the other pictures we'll see that a little bit more pronounced. So the torpedo phase, the lobes point inwards. Okay, so it's a little bit difficult sometimes to distinguish late heart and early torpedo because remember this is a continuous process. The walking stick phase is when the lobes here, the cotyledons, actually bend over. Okay, so look, it looks like a walking stick. So again, the developmental phases or the morphogenic phases all occur in the globular and the early heart stages. All of the basic tissues, all of the basic organs, and your apical meristems, your shoot apical meristem and your root apical meristem, by this time, by the formation of the heart phase, have all been established. And so once you get into the torpedo and the walking stick phases, all that's happening is growth, nutrient storage, and finally the establishment of dormancy. So let's talk about some of those features. So you need to know what are the axes of the plant body, what are the basic tissue types that are established by the embryo, and what are the organs. And for each of these, you need to know when they are differentiated. Okay, so there's two axes to the plant body, both for the embryo but also for the mature sporophyte. And the first one is the apical basal axis, right? This is the apex or the top, and this is the base or the bottom. So longitudinal, top to bottom. The apical basal axis is top to bottom. And so that's actually established very early in the globular phase when the root apical meristem differentiates. The other axis is the perpendicular or the radial axis, and that's the cross-sectional axis. So that begins in the center, and we see these concentric layers of tissue. Okay, so while when you look at the apical basal axis, you see at the apex, you see the stems and the leaves, and at the base, you see the roots. In the radial patterning, you see tissue types. So in the center, you see vascular tissue, then you see ground tissue, and then you see epidermis. All right, so just like we talked about in the female gametophyte, the zygote has polarity. And a lot of this is carried over from the female gametophyte because remember, the embryo and the zygote are developing within the ovule, and the ovule matures to become the seed. So in the female gametophyte, we had the chalazial end and we had the micropylar end. And remember that in the chalazial end, that was the closest point on the ovule to that vascular strand that runs through the funiculus. So the egg cell was located at the micropylar end, and the egg cell, once it gets fertilized, becomes the zygote. Okay, so the seed is also going to be polarized. You're going to have the chalazial end and the micropylar end. Excuse me. And as the egg cell grows and divides, it establishes the polarity of the zygote and the embryo. So the orientation of the embryo is determined before the zygote is even formed because of the micropylar end versus the chalazial end. Um, so the first thing that happens during embryogenesis is the fertilized egg or the zygote elongates along its apical basal axis. So it gets longer, it makes a long cell. And then once it gets ready to divide, it divides via an asymmetric cell division. So you get a small apical cell and a long or larger basal cell. And I need to stop now for a second, but there will be another video right below this one.